Welcome to the Jack Westin MCAT Podcast with your hosts, Michael Maleka
and Molly Kilty. Hello, everyone, and welcome back to another episode of our Jack Westin MCAT Podcast with your hosts, Mike and Molly. Mike, this is going to be a pretty interesting transition here. Last episode, we were very focused on muscles and contraction. This week, we are totally going off in a new direction.
Totally different direction. Like
Totally
it's kind of like muscles actually have nothing to do with this. Like we are
at all.
at all. But you know what? It's on the MCAT. That's how this is related. We are talking about a topic that's so important, and it's like this is what precedes muscles, really, way before that. Because today we are talking about meiosis and gametogenesis. We are breaking down, the formation of those gametes, those male gametes, those female gametes. What are the stages and the importance? Where does the weird stuff happen and how the MCAT tests it? Right? This is going to be stuff that's really important because, right, I think there's this is one of those topic areas where I think we both agree you're not going to see on every single passage, every single exam, right? It's not the highest, but it's also not low yield either. It is very reasonable to get questions on these, but we see so many errors with this, misconceptions about meiosis, misconceptions about when things happen, at what point things happen. So super excited to break that down today with you, Molly, to kind of really clarify when things actually happen during meiosis and how that corresponds to gametogenesis in humans, where the meiosis in us actually happens.
Yeah. And I feel like it's important to start us off here with meiosis as a whole, just like, what the heck is this? When do we go through meiosis? What is meiosis? What types of cells will experience this, etc., right? Meiosis is going to sound a lot like mitosis. And I think most students, at least at some point, whether it was high school or college or for MCAT, we've all learned mitosis, right? It's PMAT, right? Prophase, metaphase, anaphase, telophase, cytokinesis. It's nuclear division, technically, and then division of the cytoplasm, cell division, right? Same, the parent cell looks just like the daughter cells. It's not a literal photocopy. Obviously, it's more complicated than that.
But it's pretty close.
But when you look at the two daughter cells, they look like carbon copies, so to speak, of the parent cell. Meiosis is very different. Okay, meiosis is very specific to our germline cells, right? Specific cells get permission to undergo meiosis for an entirely different purpose, right? Whereas mitosis is just, we call it asexual reproduction. The goal is to make cells that look like you, right? To divide and replenish cells like you. Meiosis is sexual reproduction, right? Or it's for sexual reproduction. The goal is, I'm going to take a cell that is just like my other cells for the most part in terms of number of chromosomes, amount of DNA. I'm going to replicate my genome, just the same as mitosis. You replicate once and you divide once. Daughter cells are the same. Here, we're going to replicate once and we're going to divide twice, which means each of my daughter cells, first of all, I'm going to get 4, but each of my daughter cells is going to have half as many chromosomes as the original. Okay, and why, right? We'll go through the steps. There's actually a couple other really interesting parts of that process. But when you think of why, the whole point is to have 2 gametes meet and then ultimately fuse and create a brand new combination of chromosomes, right? You have fusion of gametes from a maternal side and a paternal side, right? A sperm and an egg, and you get a new zygote who has a combination of genes from both parents. And that's the goal. So meiosis is the process of generating basically distinct nuclei that are different from each other, right? Those future cells, the products of meiosis and cytokinesis, technically, are going to be different in many ways. And that's the point. It's all to increase our genetic diversity and hopefully to mix up our own genes, right? Swap genes on different chromosomes, do all this fun stuff with independent assortment and segregation and crossing over and things that we'll talk about. And then ship off your gametes and hope that if fertilization occurs, that you've passed on the best of both worlds from your maternal and paternal side. It's
Right.
kind of cool.
It's so great, and it's like why, like when two parents have a child, they have traits of both of them. It's not like
Mhm.
carbon copies of one or the other. It's a
Yeah.
mix. It's a blend, which actually holds really important genetic like importance, like in terms of evolution and biology and adaptation in order to adapt to the environment, that genetic diversity. Yeah, so like you said, so important though that those gametes that we form only have half the amount of DNA that it started off with. Because imagine if they had full amounts, that means you'd have a resulting zygote with like double the amount of DNA
Yeah.
that it showed. Way too much DNA. So no,
Fun fact, that is of common mode of speciation in plants. Just cool, evolutionary biology. We don't do that, but plants do that. They have
plant.
whole genome duplications. Plants are awesome. Okay, back to
Yeah.
you.
You can rely on Molly to
I
bring
was
in
like, hold
evolutionary
on,
biology.
wait.
You're like, well.
Plant fact.
And so that's important. So when we talk about this like halving of the DNA, this is where we talk about the ploidy of cells. Because on a big picture, right, our cells, we know we have two copies, right? That's why we call our cells, like regular human cells, diploid. Diploid means there's two N or two copies. When you see two N, that means there's two copies of the DNA. And as a result of meiosis, right, those N products, those gametes, will be haploid or 1N. I kind of think about they have half the amount of much of DNA. That's why they are haploid. So we're going from diploid cells to haploid cells. That's like the big picture, right? And though when you hear those terms of a ploidy, that's like the N number. That's how many copies of DNA that you have. And so now we can talk about the different phases because we need to go from diploid to haploid. But now let's talk about the actual process of the meiosis, about when it actually goes. And I think that process, there's a lot of misunderstanding about the process of meiosis, about what happens about when it actually becomes haploid, which I think is super
Mhm.
cool. And so when we're about to go through the different phases of meiosis, and I think Molly, when we were talking about this before, you said something super funny that I have to share. Right, when you think about meiosis, all the weird stuff happens in meiosis one. All the strange stuff. I remember it's like, yeah, the weird stuff happens in meiosis one. And that's so true. Meiosis, we're kind of looking at a big picture, has two phases. Meiosis one and meiosis two. And we're gonna go through the steps. But before we even go in, I just want you thinking meiosis one, that's where the weird stuff's happening. That's where the strange stuff's happening. Meiosis two is really gonna replicate mitosis. It's gonna be a lot like mitosis, where kind of classic, but meiosis one, that's where the weird. I just had to bring that up.
It's so funny because I just taught, for context for anyone listening, I'm currently teaching one of our intensive courses, which is like the boot camp, and I'm teaching one of our longer 515 courses. And I actually just taught this material in both of my classes, like within the last 3 weeks or so.
Mhm.
And I know a lot of our students listen to the podcast, so you're gonna hear me say this, students. Everything weird about meiosis happens in meiosis one. I say that like 100 times every time I teach meiosis because there are so many questions about the timing of like, hey, when does crossing over happen? Crossing over is weird. It's in meiosis one. When does
That's weird.
independent assortment happen? Is it weird? Meiosis one. When do homologous chromosomes align at the metaphase plate? Which, yes, I know this is a bunch of crazy language that we haven't talked about yet. If it sounds weird, it's probably meiosis one. Okay,
Mhm.
meiosis two is just like mitosis. If you know mitosis, you're fine. We'll talk about meiosis two as well, but like everything weird about meiosis happens in meiosis one, especially going from diploid to haploid, like you said.
Mhm.
I think that's the most confusing thing. When do we have 46 chromosomes?
Mhm.
When do we have 46 duplicated chromosomes? So two
Mhm.
sister chromatids per
Right.
chromosome.
Right.
And then when do we go from 46 to 23?
Hmm.
Right. The weird thing, the reason why we go from diploid to haploid in this process is we have 46 duplicated chromosomes per cell at the start of meiosis one. After that first division, instead of going 46 duplicated to two with just regular 46, like mitosis, we go from 46 duplicated to 23
Hmm.
duplicated
Hmm.
each.
Hmm.
We went from 46 chromosomes to 23 chromosomes. You go from diploid to haploid. That's why it's weird. Then you have 23 duplicated and you go to 23 each. And that's just like mitosis. So everything weird about meiosis happens in meiosis one. I know some of my students are getting flashbacks from class,
Right. Hmm.
but it's helpful, right? We're going to
Hmm.
talk about a couple weird things We're going to talk about tetrads and crossing over in prophase one. We're going to talk about independent assortment at the metaphase plate, right? The separation of homologous chromosomes in anaphase one. Everything weird about meiosis happens in meiosis one.
That love that. So let's get started with meiosis one because like going
Yeah.
into meiosis one, something we have to recognize before, and you mentioned it earlier, and I want to hammer it down. Before we've even entered meiosis, the DNA is already replicated, right? A
Yes.
lot of the times we see the DNA in its replicated form, but like it doesn't always exist that way. Like DNA is normally just a single kind of strand of DNA. All our chromosomes, if you see a karyotype, that's a term we should know, where they kind of line up the DNA by size, right? The DNA strands, like chromosome 1, is just a single strand of DNA, right? But when it's replicated in S phase, right? Now every chromosome kind of looks like an X, right? That's what it kind of like looks like. Everyone's kind of used to seeing, oh, the X, that's how DNA. No, that's the replicated form of the DNA. So now you have 46 copies, right? 23 unique, 46 individual chromosomes, but that are each X's. So you essentially, if you're talking about the individual chromatids, which are each branch of the X, right? If anyone who's watching or not watching, I'm holding up my fingers and crossing them over. Each branch of the X is a chromatid. You essentially have 46 times 2 chromatids. That's a total of math 92 chromatids. So you have a total of 92 chromatids right now, 46 copies kind of deal. So now this is where we enter this meiosis one where, okay, we've already have the replication. We have double the amount of chromosomes. And so now we enter prophase one. Meiosis is nice because it has the same names as mitosis, but we put like a 1 and a 2. So where we know mitosis was PMAT, prophase, metaphase, anaphase, telophase, we also have prophase 1 as the first phase of meiosis. And so this is the one where, kind of similar, the chromosomes condense. Because normally chromosomes are like spaghetti, right? They're like everywhere in the nucleus. This is where they kind of condense into that really nice X shape that you'll see. But then now, this is where weird stuff's happen. This is where weird things happen. So these chromosomes, normally, if we're thinking normal mitosis, they're just going to, the different X's are going to line over the plate one on one. But that's not what happens here, at least right away. What happens here during prophase one is our different homologous chromosomes synapse. Now, what are homologous chromosomes? Well, we have to recall we get one copy of every DNA strand from our mother, one copy of every DNA strand from our father. Homologous chromosomes are the same chromosome, but one from the mother, one from the father. So we know we number them 1 through 23. You have a copy of chromosome 1 from mother, a copy of chromosome 1 from father. Those copies are homologous chromosomes because they have the same genes on them, the same overall genes, but maybe different alleles. So they are not identical. They are not identical, but they have the same genes. Those are what we call homologous chromosomes. And it's during prophase one that these homologous chromosomes synapse. That's the word, that synapse to one another, or they join at the center. So now instead of 1x, or one chromosome with 2 chromatids, you now have the formation of what we call the tetrad. So now you have this kind of super chromosome, that's not formal terminology, but this super chromosome with 4 chromatids on it, all joined at the center, right there. So now you've got this giant tetrad. And with the formation of the tetrad, now these homologous chromosomes are essentially attached together. And this offers the opportunity for this kind of another weird thing that happens during meiosis called crossing over. Because before we knew, okay, we have 2 homologous chromosomes with each has its duplicated sister chromatid, right? That identical one after the replication is joined together. But now the chromosomes that are touching from the X's now can cross over so they can trade genetic material. This is an enzyme-mediated process, but they essentially trade material so that, oh, some of the genes that were on the mother's chromatid is now going to be transferred to the father's, right? So this is actually what causes, normally the sister chromatids are identical, right? But now you have non-identical sister chromatids. You've allowed for crossing over because of the formation of that tetrad, which is weird. But of course it's weird because it happens in meiosis as well.
Yeah, of course. Yes, everything weird that happens in meiosis happens in meiosis one. And I think it's so cool that we do this. And I hear so often from students like, what the heck? What is the point? Keep in mind, to your germline cells, right? The second that meiosis 1 and 2 are complete, these gametes go off and do something. It actually really depends whether we're talking about a male or female, biologically, what happens to that gamete after being produced. But I like to visualize it as like someone doing like a message in a bottle out to sea. Like, I don't know. I don't know if this is going to matter. I don't know if anyone will end up reading this, but I'm going to put my best work, my best information, and I'm just going to lob it out in the ocean and hope that it's good, right? Like, I want to put the best that I have together and just hope that luck brings this to somebody that can take action on it, right? It's like these cells, these germline cells are like, I got some good stuff from my maternal side and from my paternal side. I want to make sure that I have gametes that have the best of both worlds. I'm going to let them switch, right? I'm going to switch things around a little bit so that I have a better chance of maybe accidentally taking the best of mom's side and best of dad's side and giving those both to one of those cells. It's so clever and so cool that we can do this. And again, the whole goal is to increase genetic diversity, to basically give the opportunity to have many different types of gametes that are produced, which is why, if you think of siblings, like, it's funny, I have 4 siblings. I'm the youngest of 5 kids. I don't think that me and my siblings look that much alike. Like, no 2 of us look identical. And yet when you look at us all together, we all look similar.
Like,
Like,
you could
all
tell your
5 of us.
siblings.
You could tell we're all siblings.
But if I like picked up 2 of you guys, by the way, I haven't actually
Exactly.
seen Molly's siblings before. But if you picked up just 2, you'd be like, oh, you guys don't really look that much alike.
Yeah, any 2 of us don't really, but when you see us as a group, it's like, oh, okay, well, you guys kind of have the same eyes, and like you 2 have the same nose, and you have this same feature. Like, we each share different things, and none of us really look the same. And it's because while we both started with my mom and dad's genome, we got all entirely different combinations of them. And it's because of things like prophase 1, right? Crossing over. It's actually also going to happen in metaphase 1, our next phase. When we leave prophase 1, right, we formed those tetrads. We have the 2 homologous chromosomes that have undergone crossing over and exchanged things. They're still snuggled up pretty much next to each other, right? When we talk about metaphase and mitosis, we know we're going to align at the middle, right? We're going to have those kinetochore microtubules extend from our microtubule organizing centers or our centrioles. They're going to extend and they're going to attach. And then we're going to, for metaphase, just stay there. Anaphase, we're obviously going to cut them and we're going to separate. But metaphase is, we're aligned in the middle of the plate. We've got microtubules attached and we're ready for the next phase. With metaphase 1, right? Everything weird about meiosis happens in meiosis 1. Something is unique here, right? Because normally every single or both of your daughter cells are going to get 1 copy of mom's chromosome and 1 copy of dad's, right? That 46 duplicated to 46 each, period. That's how it works. Here, though, we're going from 46 duplicated to 23 duplicated each. So one of those, or each of those daughter cells of meiosis 1 are going to get some of mom's chromosomes and some of dad's. What happens at metaphase 1 is we basically say, it doesn't matter. There's no rule of like, when we think of those 4 chromatids, right? The tetrad, there's no rule that like, oh, mom's chromosome always has to be on the left and all of dad's have to be on the right. There's no rule. You mix and match. It's random, right? And that actually creates a lot of different options, right? Gamete 1 might have, let's say, 10 of mom's original chromosomes and 13 of dad's, and vice versa in the other gamete, right? It could be, 23 and 0 and 23 and 0. Like, there's so many different ways that you can have these tetrads align. Mom's could be on the left, dad's could be on the right, or vice versa. And you have 23 different places that have 2 options each. There's actually an equation, not really a full-on equation, but a mathematical expression that we use for this. If you want to figure out how many ways you could align at the metaphase plate, we say it's 2 to the n, where n is the number, or it's your haploid number. So like we have 46 chromosomes, 23 unique sets of genes, but of course you have a mom copy and a dad copy, or a maternal and paternal side. So N for us is 23. So we have 2 to the 23 different ways to organize mom and dad's chromosomes at our metaphase plate. And that is a lot. And it looks like you're typing. Are you getting
I literally was like, oh yeah, that's about 8 million different combinations
Yeah,
over 8 million different ways
it's crazy.
that different types of gametes that could come out from there. Right.
And that's only metaphase. That's
Mhm.
not even counting the additional genetic diversity you get in prophase with crossing over. So
Mhm.
that's huge. This is again how I could have 4 siblings and none of us look identical. Um, which I need to show you a picture of my siblings now that we've talked about it so much.
I'll be like, yeah, you guys look nothing alike.
Yeah, I know. We're all tall, and that's pretty much it.
That's what we got in common. That's it. It's so funny
Yeah.
because I have one younger brother, and we are like very like people can tell we're siblings. Like it's like
Oh,
very much
really?
like when we were younger, people thought we were twins, which always annoyed me because I'm like I'm the elder brother, like I look older. But like now that we're a little bit older now, like we have differences. It's funny. I had a For anyone who's ever watched the video, I have facial hair. My brother used to not to, and it was very easy to separate us. But then he started growing facial hair. Now we look together. Like now we look identical again. Now it's just the worst. But anyway, I'll show you a picture of my brother after. You maybe will be able to see.
Yeah.
Um, so we know. Coming back to enough about our siblings. Now that we're back on, so we've done metaphase one. They're lined up, kind of. It could be random, which whoever mom's on the right side, dad's on the left. It doesn't. There's no rule to it. But now, this is where anaphase one happens. And anaphase is where the actual splitting occurs. Now, this is super important what happens here and why weird stuff happens in meiosis. And I kind of have a rule. Right? In mitosis, we expect mitosis, 2 identical copies because sister chromatids are being separated. Right? So when sister chromatids are being separated, the same amount of ploidy is kept. Split sister chromatids. Okay, so that means you still say diploid. That means you still have 2 copies each. However, in anaphase one, again, these aren't chromatids that we're separating. These are homologous chromosomes that are separating. So because we're separating homologous chromosomes, not sister chromatids, it is during anaphase one that we go from diploid to haploid. Such a common misconception is to think, oh yeah, it goes diploid to haploid in the second phase of meiosis. But again, this is weird. So it happens during meiosis. Again, I love that. I give all credit to Molly for that one, but I love that. Yeah, it's weird to go from diploid to haploid. So that means it happens during meiosis one. It's the first time. And so now, while we're separating, that means each centriole is pulling towards it 2 sister chromatids together, right? But that means it only has half the ploidy, right? This is where it becomes haploid, right?
Yeah.
And so idea is so important that happens here. Another thing that might come up here though is when things go wrong, right?
Mhm.
This is where a term that we need to know about nondisjunction occurs. Because something might happen, as all things do in nature, where maybe those homologous chromosomes don't separate. Where instead of separating the 2 homologous chromosomes, one homologous chromosome one way, one the other, it might be that both homologous chromosomes goes to one end. So one like a cell will get both the mother and the father, one will get none, right? This is what can lead to like issues down the line, like trisomy and monosomy, which we'll discuss a little bit later on. But it's because of this lack of separation during anaphase that we get non-disjunction.
Yeah, and non-disjunction is going to be interesting to come back to with meiosis 2 as
Hmm.
well, because you actually can have non-disjunction happen there too. Technically,
Hmm.
you could have non-disjunction happen in mitosis. It's just, hey, we didn't separate chromosomes like we should have. But what that would look like is very different depending on when and where it happens. So this will be fun. Really, after anaphase one, telophase one is very similar to regular telophase. You basically undo prophase. So
Hmm.
you just reform the nuclear envelope. You're doing your normal stuff,
Right.
right?
Right.
You're decondensing your chromosomes. You're finally actually solidifying. Yes, I have formed 2 different nuclei. You have cytokinesis. Great. We divide into 2 different cells. Again, those 2 daughter cells, unlike mitosis, are not identical to the parent at all. Very different. They're
Hmm.
haploid. They have unusual combinations of alleles from mom and dad through crossing over. It's very unique. Everything weird about meiosis happens after meiosis one. or in meiosis one. So after we have two daughter cells that are both haploid, but have both sister chromatids. Okay, moving into meiosis two, really, there's nothing weird about meiosis two. It was weird. It would have been meiosis one. So when you think of meiosis two, it's literally mitosis, except that you started with half as many chromosomes. So now, again, we went from 46 through S phase, we became 46 duplicated. So you have the X, right? Still connected at what I think of as like the belly button, right? Like you're still connected at the middle of that X. And now we went from 46 to 23 duplicated. Meiosis 2 is just taking those 23 duplicated chromosomes with two chromatids each and separating those both to separate gametes. Okay, so that's it. It's the same process, except that, again, you have half the number of chromosomes. And technically speaking, mitosis, your sister chromatids are 100% genetically identical. Because these are coming from meiosis one, those sister chromatids may not be entirely identical because of crossing over, but originally they were. Again, the weird thing happened already in meiosis one.
This is just the result of the weird thing.
Yeah. So we're gonna align sister chromatids at the metaphase plate. No crossing over, nothing weird in prophase one. So metaphase two, we're aligning at the metaphase plate. We're gonna divide the sister chromatids during anaphase two. Telophase two is just gonna be the same thing, and you're gonna go through cytokinesis and you're done. Okay, that's it. So 23 duplicated to 23 each, and you call it a day. Now, you brought up nondisjunction. Nondisjunction can also happen here. What I think is cool is when you compare nondisjunction in meiosis 1 versus meiosis 2, the question is, which one is worse? Like, which one creates a greater impact on those four gametes that you do? Like, and which one would you think? The earlier problem or the later problem? Just speaking logically.
Logically, the earlier one, because that means it affects everything downstream, right? The weird stuff happens. That's when it affects everything after. So 100% the first one.
Yeah, right. And if you think about it, if you have one cell, 46 duplicated chromosomes, and you end up with two daughter products from that, right? And we know they should be 23 duplicated chromosomes each. But what if you mess up, right? In that first division, when you're aligned at metaphase one, in metaphase one, at the metaphase plate. Like you said, what if you don't separate those homologous chromosomes well? Well, now you have the first pair of daughter cells. One has 24 chromosomes that are duplicated, and the other one has 22, right? Now, each of those are going to divide. Okay, the one that had 24 duplicated chromosomes, now those daughters, right, are going to have 24 each. That's an extra, right? The one that had 22 creates 22, right? That has one too few. If you mess up in meiosis 1, all four of your gametes have the wrong number of chromosomes. Whereas if you mess up in meiosis 2, you already have two daughter cells. Each one of these daughter cells is going to divide. Okay, so maybe you have two normal, 23 duplicated chromosomes each, and maybe one of those two has an issue, right? Maybe same deal, one gets 24 and one gets 22. Okay, 2 out of 4 of your gametes are going to have a improper number of chromosomes, but the other one is probably fine. Okay, so you have 2 affected gametes and 2 normal gametes. So again, you have to think, if we have nondisjunction, when, right? In mitosis, if you had nondisjunction, you aren't sending those off for fertilization and to create a new organism. So if you had nondisjunction and it created an abnormal functionality in that cell, it probably just undergo apoptosis and call it a day.
It happens regularly all the time.
Yeah, and it's fine, right? The cells know how to respond to that. This is more important in many ways with nondisjunction because these, again, it's like a message in a bottle. You're just sending it off. You don't know what's going to happen. It has the capacity to create an entirely new organism, right? A lot of our products of nondisjunction, those gametes, oftentimes, if you have an improper number of chromosomes, it actually creates a non-viable offspring, right? Maybe you have fertilization, but that offspring cannot survive. There are quite a few trisomy disorders, right? Or, different variants of the human experience. Trisomy 21 is very common. That's Down syndrome, right? Interestingly, there are kind of quite a few trisomy situations or disorders that you can have where you have 3 copies of a chromosome, but there's only one time that you could have a monosomy. There's only one option.
Is that what the sex chromosome? Yeah, that's what the sex chromosome. I was like, wait a minute. I was like, there's no viable autosomal monosomies. That's where you got me. I was
Yeah.
like, I know where you're going with this. Yes, yes, yes.
I mean, in a way,
Mhm.
Mike, you have a monosomy situation.
Oh, you're not wrong. You are not
You
wrong.
you have one copy of an X chromosome,
Mhm.
right? Males are XY. You have a Y chromosome. Technically, you
Mhm.
have a pair of sex chromosomes, just like females that have XX, but you have one of each. And Y, as we know, is pretty tiny. It really
Yeah,
doesn't
yeah.
it doesn't really give much,
Mhm. It gives a little
except
bit.
the
Yeah.
SRY, right,
Hmm.
which keeps you or which converts you to a male early on.
Hmm.
But
Hmm.
really, you are going about life with one X chromosome,
Hmm.
right? Females have two, but it is actually possible to not have XY, but to just have X.
Hmm.
That is possible. There are
Hmm.
people that have just one X chromosome, and it's not really that big of a deal,
Hmm.
right? They don't have that SRY gene, so they remain female, right, as in terms of development.
Hmm.
But it's an interesting situation because,
Right.
as we've talked about in the past, females have an interesting mosaic situation going on. If you're like, ooh, that's interesting. We did a whole episode on
It's
genetics.
so cool.
It's
It
a
is so
callback,
cool. Yeah.
but
Check out our episode on genetics because the idea of like the whole mosaic of the females, I think is such a cool
it's so
idea.
cool.
Such a
And
cool
we got to
idea.
explore it in a
Yeah,
lot of
so
detail
you
back
should
then.
you should check you should anyone listening should check that out. But yeah, no, so that's actually the cool like the sex linked ones, they you can have viable monosomy, but there are no viable autosomies, right? Autosomal
Mhm.
traits. And so but this is a result, like we kind of bring you back to that non-disjunction where if you have one
Mhm.
more or one less, it pairs with one that has 23, 24 plus 23, that's one extra. 22 plus 23, that's one less. So that's a monosomy where now you should have two copies. That's the kind of the normal experience, the regular experience. Then you only have one trisomy. Instead of two, you have one extra, right? So that's three. And so this is kind of like a breakdown of meiosis where we kind of went through the steps. What were the kind of really important things to know? Kind of in summary, like the things that are, I kind of really want to hit on the differences between
Yeah.
meiosis and mitosis, the formation of the tetrad. That's big, right? The formation of this crossing over via the chiasmata. I don't think we use that term, but like that, those points of crossing over, where the crossing over happens, are called chiasmata, right? Where that actually crossing happens. Then the separation of homologous chromosomes, that never happens in mitosis, but that is unique here in meiosis because that's what turns diploid to haploid, right? And these non-identical sister chromatids. Everything else, though, pretty mitosis-esque.
Yeah.
Mhm.
Yeah.
Mhm.
That's a good summary, right? Just the
Mhm.
things that create genetic diversity, those are the main things to know. Again, two
Right.
divisions, that's weird, but crossing over,
Yeah.
independent
Yeah.
assortment, all those good things, right? Everything weird happens.
This is,
This is
this is,
one.
I think it's time to talk about. So we've talked about meiosis, right? We've maybe covered it before, but now let's focus on human meiosis.
Yeah.
Where does it happen in the humans? And this is where we talk about gametogenesis, right? This is where we want to talk about. Let's talk about the specific cells because between biological males and biological females, it presents pretty differently. And
Yes.
I like starting out with the male one because I think that one's far more simple. Like
Easier.
way easier. Like
Yeah.
it's so much more straightforward. Because I think it's very like there's not much variance. Like if you know meiosis, you get a pretty good idea of spermatogenesis. Now,
Mhm.
spermatogenesis is where meiosis embodies in males, where the gametes that we form are what we call the spermatozoa. They're at the ends. But let's start at the beginning, right? First, spermatogenesis happens in the seminiferous tubules of the testes in males, right? And these cells that start, these two end cells, these diploid cells that start off spermatogenesis are not sperm, right? It's not gametes that go through. This is a common thing. And actually, I
Yeah.
remember vividly making this mistake, right? When I was in like high school biology, I remember getting something wrong. Again, notice how vivid that was because I said it was gametes that undergo meiosis. No, it is not gametes. Like I've made that mistake and I never made it again. No, no. Gametes are the outcome of meiosis, right? The cell that actually goes through meiosis in the male is called the spermatogonia, right? So it's a spermatogonia, is multiple spermatogonium. That is the kind of pre-sperm cell, the cell that will undergo meiosis. And so because it hasn't gone through it yet, this is a diploid cell. That means it has two copies. And so this is the one, though, it is going through mitosis regularly. So in males, we are constantly going through meiosis. Like it's like a daily cycle. It's happening all the time. And so we're constantly replicating these spermatogonium, right? Constantly producing them. But the first step, which is the meiosis one step, is, oh no, sorry. I should go back. I kind of got ahead of myself, right? They kind of have to mature a little bit into the primary spermatocyte. This is the one that will eventually go through meiosis. So primary spermatocytes are diploid, right? Once these primary spermatocytes are ready, it's kind of like it goes from spermatogonium to primary spermatocytes. These are the ones that will undergo meiosis one to form the secondary spermatocytes. Remember, all the weird stuff happened in meiosis one. So if the primary spermatocytes are 2N, the secondary spermatocytes are haploid. These are the ones that are haploid because the first meiosis has happened already. Now, these secondary spermatocytes will undergo meiosis two. There's no pausing, they just proceed through it. Again, you'll know why I'm prefacing that now, later. But it just keeps on going. It goes like spermatogonium. All right, now it kind of gets ready for meiosis, primary spermatocyte. Now, secondary spermatocyte, we're haploid. Then it goes through meiosis two, and we form spermatids. These are also haploid, but these are not mature spermatozoa. These are kind of the immature spermatids. They're haploid, but they're not quite ready. They're not quite mature, right? They go through a maturation process in order to become the spermatozoa or the mature sperm, right? So this is the process, but each one corresponds to a phase of meiosis, right? Or so primary to secondary, meiosis one, secondary to spermatid, meiosis two, and then spermatid to spermatozoa is just that kind of after maturation. When it comes to males, this process of spermatogenesis starts at puberty and happens for life, right? Once males hit puberty, right, we get those like male characteristics, right? That development. This is when it starts, and it goes on essentially till end of life. We're constantly producing more sperm. They will die. We'll produce more sperm. There's no phases in that way. We kind of do it on a daily basis, right? And when we're talking about the spermatogenesis, these are kind of the cells, but we do need to be familiar with like in the testes, in the seminiferous tubules, there's kind of two supporting cells you need to know about. Right, first are the main cells that aid in spermatogenesis, which are the Sertoli cells. These are cells that are within the seminiferous tubules, and they are there just to support, which is via supplying nutrients and signaling molecules that the MCAT doesn't really focus on, but you just need to know that they kind of just help spermatogenesis along. These Sertoli cells are critical for them. But another cell that's also very important are the Leydig cells. Now, when people think, what do the Leydig cells do? The Leydig cells are what actually produce the testosterone in males. I think most people know, like, testosterone comes from the testes, but the cells that actually produce the testosterone are the Leydig cells. It's kind of like in the pancreas, people are like, insulin glucagon from the pancreas. They're like, no, well, it's the alpha and beta cells from the islets of Langerhans that actually make the insulin glucagon. If what I just said was like, oh my goodness, hormones, go check out our endocrine episode. But these are where, so our Sertoli cells help spermatogenesis, the Leydig cells are what produce the testosterone. And we can kind of go back and like, okay, which hormone actually leads to the activation of these cells? So for the Sertoli cells in males, it is the follicle stimulating hormone. Right? A lot of the times that people think of the FSH and LH solely in females. No, males have FSH and LH too, and these serve very important purposes in males. FSH target the Sertoli cells, critically important for this process of spermatogenesis, where the LH, the luteinizing hormone, is what corresponds to Leydig cells and what leads to testosterone production. I always remembered like the L's match up together, LH and Leydig, but FSH, yeah, that's just how I always, because I always mixed up which goes where. But yeah, the L's match up in males. But FSH, important for the Sertoli cells, which aid in this process of spermatogonia to spermatozoa. So, man, I just rambled on for a while. Oh my goodness.
But you gave us the entirety of spermatogenesis, which is awesome. And as the one who's going to talk about oogenesis, I apologize in advance
Yeah.
because it is an even more complicated
It's
process,
so much more.
right?
It's like that, if that was you thought that was bad, just wait until
I mean, it's better and it's worse. It's just unique, okay?
Mhm.
I will say it's good to go over spermatogenesis first because you're gonna see the same same suffixes for
Mm.
the spermatogonium, primary spermatocyte, secondary spermatocyte, etc. that we're gonna see here. Okay, but of course it's gonna be oocyte instead. So let's talk about this, right? Switching to our biological females out there. This process of gametogenesis, it's called oogenesis. It happens in the ovaries. Okay. And we start with oogonia or oogonia, however you want to phrase it. Okay. And this is the female version of a spermatogonium. I always like to think that oogonia remain in the gonads. It's got the go.
Mm.
Okay, it
Mm.
stays there. These are the stem cells, right, that ultimately divide a lot by mitosis to produce a lot of I almost said primary spermatocytes, but primary oocytes, right? In our case, or in the case of biological females, right? And this in I'll go through just the major phases and then I'll talk timing because it gets a little confusing, right? The names and the phases are the same. So you have oogonia, right? That ultimately will divide by mitosis. They remain in the gonads. They produce primary oocytes that are diploid. Okay, and these are the ones that are actually going to go through meiosis one. I always remember this. Primary spermatocytes or oocytes are going to go through meiosis one, right? They're named
Mm.
for what they're about to experience. So they're primary because they're going through meiosis one, and they create secondary spermatocytes or oocytes because they're about to go through meiosis two. Okay, so we are diploid with our primary oocytes. Now we are haploid, but duplicated with our secondary oocytes. And then we're going to go through or finish, right? We're going to complete meiosis two and create ootids or just eggs. Oftentimes
Mm.
they're
Mm.
described.
Mm.
Because we're going to talk about how it's very different, right? Spermatozoa, right? They actually leave the body and go on this long journey, which we will talk about later in another episode. They actually leave the body and have to cover a lot of ground, very literally, before they are ready for fertilization. Oocytes, not so much. The
They're
timing of
chilling.
this is actually really funny. I think it's hilarious. So let's talk about timing. Name-wise, very similar. In terms of what it looks like, actually quite different. And in timing, very different. Okay, so we actually undergo that initial division and maturation process, right? The oogonia to the primary oocytes before we're actually born. Okay, so there's this common thing told of like, oh, you're born with all the eggs that you'll ever have. And it's like, well, yeah, kind of. They're not really eggs yet, but you have primary oocytes that are present at birth. But these are primary. They're going through meiosis one. They're actually frozen in the process of meiosis one. They're frozen in prophase of meiosis one. Okay. And they will not do anything until puberty. Okay. So they're just going to sit there, right? Frozen completely until puberty. And at that case, right, where in males, you just keep going. Starting in puberty and basically until death on a daily cycle, you're just a sperm factory, spermatozoa factory. Here, it's very specific with the timing. Typically, typically one of those primary oocytes is going to complete meiosis one each cycle, right? So each month, females are on a typically monthly hormonal cycle, and they are going to complete meiosis one and become a secondary oocyte. Typically, one, there are times you can release multiple that can lead to dupe or double fertilization events. You can get twins that way, one of several ways. Typically, you get one. That secondary oocyte is going to remain arrested in metaphase two, actually, until literally the sperm has fused with the egg, which is so funny, right? Like we were frozen in prophase one, and then we become frozen or arrested in metaphase two, and we, as speaking as a secondary oocyte, we will not complete that meiosis two process until literally we have a spermatozoa's nucleus at our front door, so to speak. We will not finish getting ready until our guest has arrived,
Yeah,
basically.
which is so wild. I remember I have seen so many resources to be like, oh yeah, it's the ovum that's released from the ovaries into fallopian tubes. That is incorrect. It
Yeah,
is a secondary
secondary
oocyte that's
oocyte.
released into those fallopian tubes. And it doesn't
Yeah,
even, it's like it waits until like the sperm is there and it's like, okay, I guess now I'll become an ovum. So it's like
yeah,
the sperm's already there when it becomes an ovum, and then that's when like I think that's such a cool idea. And but again, not something that like people overlook that all the time. They're like, oh yeah, it just
yeah.
finishes and then it fertilization happens. Like, not really.
Yeah.
Mm.
Yeah. It's so interesting. Same thing with like, oh, where does fertilization happen? Which we'll talk about actually in a later episode. Everyone thinks that the oocyte is released directly into the fallopian tubes. It's actually not.
Mhm.
Fallopian tubes got those
It's
little little hairs.
in like this open space, and they're like, I guess I'll go. It's
Yeah,
wild.
yeah.
Yeah.
Anyways, super
Mhm.
cool. We'll talk about this too. The other thing though, on the same wavelength of oogenesis, the only other thing I want to mention here, I'm saying this is like, hey, we go from primary oocytes to secondary oocytes, plural, and that's actually incorrect to say. Okay,
Mm.
the other really weird thing about oogenesis is that you only really make one functional egg. Okay. Keep in mind, meiosis is the division of the nuclei, the same way that mitosis is the division of the nuclei. Okay. And it's true that you are going to take that original cell and end up with a daughter cell that has half of the genetic material. That's true. However, there's only one functional ovum created. Okay. And that's the weird thing. And it comes down to fertilization. The reality is, pretty much everything that's needed, not just for that initial zygote cell, but as we'll see in early fetal development, or early embryonic development, really, as soon as that thing's fertilized, it's going to undergo cleavage. It's going to create multiple daughter cells. This ovum is huge and it's packed with proteins and nutrients and organelles, basically enough stuff to be divided into multiple daughter cells pretty immediately after fertilization. So these are very resource intensive to make. Okay, which is also why we biological females cannot, have this process happen every single day like biological males. It's so energy intensive to produce these. Every time we divide in meiosis 1 or meiosis 2, we make one viable daughter cell, right? That might go on to become an ovum, and one what we call polar body, which is basically just a nucleus. Like technically it's got its own, it's like a cell, but it's got really nothing else that's helpful, right? It doesn't have
Mhm.
enough organelles to support it. It doesn't go off and have its own life, really. It's just kick out the unused nucleus and focus on the giant resource intensive cell. And of course, that's going to happen again. So you really produce one functional ovum from
Mhm.
that, and then a couple polar bodies. So
Yeah,
that's
and
the other very weird thing
yeah,
about oogenesis.
which is again so different from spermatogenesis because spermatogenesis is very much you get four spermatozoa, they're ready to go, all equal amount of nucleic information and like very little of anything else really. Like all the sperm really gives to the zygote, to the eventual zygote is the nucleic information. They give like
And
really
a centriole. Don't forget
and
the centriole.
essential, I should be, yeah, and essential to help, absolutely. But like in terms of like ribosomes, mitochondria, like all these things, like they give like nothing else. Whereas it's really the oocyte that's really giving everything, and that's why, right? You're just going to form these polar bodies. But a common MCAT kind of trick is they always ask about like, oh, the difference between the polar body and like that oocyte that forms, right?
Mhm.
Oocyte gets all the stuff outside the nucleus, right? But when it comes to nucleic information division, both the polar body and that spermatocyte get the same amount of nucleic information, right? It's not
Mhm.
like the that oocyte that gets all the stuff outside also gets more nucleic information. No, no, it's still the same amount,
Mhm.
right? That goes in each, but it's just they get all the stuff like in the cytoplasm and that sort of
Yeah.
thing. That all goes. And I think that's also a really important idea when it comes to like who donates what. When we think of like maternal mitochondrial inheritance, right?
Yeah.
Like when it comes to like we know every one of our cells has a mitochondria, right? We know that, right? Um, However, like there are mitochondrial diseases, mitochondrial linked diseases, because the mitochondria has its own little genetic information. That's really, really cool.
Because of
Because
evolutionary biology, might I mention,
because of evolutionary biology,
and
but
the symbiotic theory.
but this idea that because it was on the male only donated like one half of the genetic information and a centriole, right? You didn't get any of your paternal mitochondria. All of your mitochondria came from your mother because that's where the oocyte was. So this kind of has like a really cool link to genetics when it comes to like mitochondrial inheritance. This is the why behind maternal mitochondrial inheritance because they get all of that. It's the oocyte that provides all the stuff other than a centriole and half nucleic information, which I think is a cool link to the why.
Yeah,
Mhm.
absolutely.
Mhm.
And I know that going over this, I feel like it feels like we've covered a lot of material, and it's because we kind of have.
Mhm.
Hopefully, we made it kind of fun. I feel like
Mhm.
this material is often like, oh, I just have to memorize it. Like,
Yeah.
it's kind of cool, to be honest
Pretty
with
cool.
you. Like,
Mhm.
it's cool. It's interesting. I like it a lot. Maybe that's just me. But there's a lot to track, and I think it's important when you're answering questions on things like this that we establish what are the criteria I'm looking for in answers. Okay, so
Mhm.
like if you get a question and it seems like it has something to do with meiosis, okay, or gametogenesis, first question you should ask yourself is, what do I have in this cell that I'm asking about? All right, if we're asking about a daughter product of mitosis or a starting material as let's say a primary spermatocyte, it's like, okay, what is going on? Okay, what is going on with this cell, right? Is it haploid? Is it diploid? Is it duplicated? Does it have two sister chromatids per chromosome, right? Where is it in the cell cycle? Has it already undergone S phase? Is basically what we're asking. If it's haploid, okay, it's probably either a product of meiosis, done, or it's at least a product of meiosis one. Okay, it could
Mhm.
be halfway through meiosis or it could be a final product of meiosis. But again, are we duplicated? Are we haploid? Are we diploid? And do we still have two sister chromatids? Right, that will help us figure out, are we going through meiosis? Are we going through, or meiosis one or two? Am I going through mitosis? And also contextually, what kind of cell is going through this? Do you have any information that this is a germline cell or that this is one of our Go cells, spermatogonium, oogonium? Those are all indicative of meiosis. I do want to mention though, if it doesn't give you a hint about meiosis in some way, it's mitosis. Like
Yeah.
vast majority of your cells will not go through meiosis. Pretty much in air quotes, every cell undergoes mitosis. There actually are exceptions, but if a cell is going to divide 99,000 times out of 100, whatever, it's
That was
mitosis.
why you use 1,000.
I don't know. I was going to say 99 times out of 100, but I'm like, no, actually, I bet it's even more than that. Like, just keep making the numbers bigger. 999,999 times out of 1 million.
Yeah.
It's mitosis. I actually have no idea if that statistic is accurate.
That's not
As
well.
you can tell,
They'll
I'm
always
making this up, but
they'll always give you like a hint, right? They're not going to just expect you with that hint. Like if they say anything about, oh hey, a germ cell was formed, or
yeah.
this is this is related to the gonads, and there's the Sertoli cells were involved in this thing, or hey, FSH was involved in this. Like anything that kind of directs you to it, you're like that's the priming. When we talk about like science passage strategy and like
Mhm.
priming is a big thing. Look for these things in the passage. If they mention anything about this, you're now primed to think, okay, meiosis one, meiosis two, testes, FSH, LH, all the weird stuff in meiosis one. All that priming happens in the passage to kind of give you a hint. Is this is how I should be thinking?
Yeah.
Mhm.
I'm so excited for this unit. It's so
Yeah,
much fun.
yeah.
There's so many weird things going on, right? So
Mhm,
if you get specific clues, right? Use
mhm,
that spreading activation we talk about in psych soc, right? Use it. It seems like we're talking meiosis. I need to keep that in mind. I don't feel like we're specifically mitosis or meiosis. It's probably mitosis. Okay,
mhm,
so lots of specifics to help you figure out which type of process you're going through, and then also just pay attention to specific words. Right? Like
mhm.
if they're asking how many chromatids are present, that's a different question from how many chromosomes are present. Again, let's say after meiosis one, you have 23 chromosomes, but each of them has two chromatids. Okay. So it's confusing, right?
Hmm.
Make sure you focus. If it's 23 duplicated, you have 46 total chromatids, even though you have 23 chromosomes. There are lots of diagrams that will visualize this.
Hmm.
Highly recommend checking it out, especially if that idea feels really abstract to you. I think that's probably one of the most complicated,
Right.
like specific questions that you want to be prepared for.
Hmm.
Other than that, I feel like it's a combo of cell cycle stuff, meiosis, mitosis, and genetics, right? And this is the basis for Mendelian inheritance and genetics and dominance relationships is you have two copies, one from mom, one from dad. They have a little war going on, right? Are they dominant? Are they recessive? What's going on? Controls the phenotype, right? It's such an interesting process, and there are so many different layers to it that we could look into. Um, I'm actually very excited for our next episode too. This is
Hmm.
like the logistics of meiosis and what it looks like. But as we've kind of alluded to here in the last little segment with oogenesis, it gets complicated. And
A little
I'm
bit
very
tricky.
excited. I'm very excited, especially as a lady myself, to get into our topic for next episode because it is one of the most hated experiences for many women, but also feared concept categories within
Hmm.
the MCAT for both biological males, females, anything in between, right? Like it
Hmm.
nobody likes learning about this except me, actually. I think it's kind of cool.
I thought it was pretty. I think it's pretty good. As a biological male, I think it's interesting.
Okay, there we go.
As a little bit someone who's a bit removed from it, but I always it's so funny when I teach like a class, like when I'm teaching about the menstrual cycle. And people
Which
ask me
is our
like,
topic, by the way.
wait, oh my goodness, yeah, you figured it out. But yeah, next episode we're talking about the hormonal control of menstrual cycle. Whenever I teach this topic, people are like, "Oh, do you feel awkward as a male teaching this?" I'm like, "No, this is so
Oh,
interesting. What are you talking about? This
yeah.
is so cool to actually understand the process." Because that's why I think it's intimidating, right? Because we feel like we have to memorize, like, "Oh, during this phase, this happens here," and oh, I have to keep all of these timings in my head. But no, how we're going to tackle it next episode is we're going to have you understand it. Why is it these things happen? When you see an LH surge, why did that even happen in the first place? What caused that thing? I'm excited to, as a biological male, to talk about that next episode.
Yeah. So we're going to be talking about the menstrual cycle and how the menstrual cycle is actually not just one cycle, how it's actually
Right.
two cycles that
Hmm.
interact, and there's all this interesting hormonal applications. If you haven't heard our endocrine episode, definitely check it out.
Really
Um,
important.
it's very important, especially when we talk about positive and negative feedback control. But we'll save that for next episode. For now, I hope you guys have a wonderful rest of your day or week or weekend, wherever we're
Wherever we
catching
find,
you.
yeah.
Yeah. Happy studying. And we'll catch you in the next one. Thank you for listening, everybody.
I know, everyone.
Bye.